So welcome everyone. My name's Nick Fuggle. I'm an associate professor in rheumatology and an honorary consultant rheumatologist down at the University of Southampton, and I've got the privilege of co-organising this group together with Emma, Alicia, and Joe. So welcome to this October edition of the Clinical AI Interest Group. A few notices before we begin. And actually, this is a couple of months really packed with activity. So obviously, we've got our meeting today. Then tomorrow on the 15th at 12:30, there's the Anesthesia and Critical Care Supra Interest Group meeting. Our Supra Interest Groups meet around particular areas of clinical expertise. And so that's for anaesthetists and those involved in critical care. Then on the 28th of this month, we've got the Genomics AI Super Interest Group Network meeting with Dr. Felicity Locke and Dr. Ankit Tanna, who I think we can call a friend of the show, both from Hardian Health and talking about the regulation of AI in genomic medicine. Then on the 26th of November, we've got the AI for Women's Health meeting with Professor Jane Hurst. Again, really exciting to see, that the super interest groups flourishing in terms of the activity they put out. And in our November clinical AI interest group meeting, we're being joined by Jeff Hogg from the University of Birmingham, who's going to be talking about an AI readiness checklist. Now, I mentioned our super interest groups earlier. I'm really excited to say that the dermatology super interest group has launched, led by Mike Arden-Jones, Roberta Matten, and also Teresa Sakok. And I can see actually from those that have got videos on that you've all got incredibly good skin. So I imagine that there might be some dermatological expertise demonstrated here. But if you do have dermatological expertise and you want to get involved, then please reach out and contact those super interest groups. And you can see information about all of those on our webpage as well. The last thing I was just going to say is that Professor Jeremy Wyatt, I think who we could also call a friend of the show, is hosting an online conjoint experiment, which I think is a type of survey, and particularly looking at drafting some AI purchasing guidelines. So information and links regarding that are available through the Clinical AI newsletter. So with no further ado, Catherine, are you still there, or have you disappeared?
No, I'm still here.
You're still here. Great, Catherine. So lovely to meet you. Would you mind telling us who you are, where you come from, and how, in some way, you interact with artificial intelligence?
Yeah, so my name is Catherine Lamriddle. I'm a senior technician at the Health Technology Hub at the University of West of England in Bristol. We're currently working with NHS England on AI projects via it's called CODE, which is Centre of Digital Excellence. So I was just coming along for a kind of a bit of a see the broader picture. That was why I was coming along. I was like, oh, this is a very small
Fan
group. I wasn't expecting that.
Fantastic. Well, huge round of applause for Catherine because that was completely improvised and we really threw that at her. But thank you so much. You coped incredibly well and welcome to the community.
Thank you. Thank
So
you very
with
much.
that, I'm going to hand over to Alicia for our thematic session.
Thanks, Nick. Catherine, I'm over the water. I'm in Cardiff, just outside Cardiff, actually. Absolutely delighted that Steve has come to join us this afternoon over lunch to talk to us about digital twins and his work specifically in cardiology. Prof. Steve Niederer is the chair of biomedical engineering at Imperial College and one of the co-directors for health at the minute in the Turing, and has been leading the way in clusters in digital twins. So Steve, over to you. I haven't got your slides, so you're driving your own slides.
Excellent. Huge responsibility, and so we'll see how we go.
Wonderful.
So have we got this?
It's coming up now. Brilliant.
Excellent. So I want to talk about digital twins, which for us are mathematical models or computational models that represent a patient, and ideally, kind of in our case, it's often the heart, and that are going to track these patients through time with updated measurements as they're collected. And it's recognizing that this is very much a journey that we are on. And so a lot of the work that I present is how we're kind of building the models, what it means to make the models. And then towards the end, I will talk about how we are applying these models to track patients individually. And for me, one of the ways of thinking this was about this idea of a puzzle, and that when we see a piece of information about the patients, we get just one piece or two pieces, and that by having this mathematical framework, which provides a strong prior or kind of a reference heart model, it provides a kind of a picture that we can have, that we can start to use to anchor the different puzzle pieces together to build this picture of the patient's heart and understand what's wrong with them. And so when we're building these computational models, we have a model of a patient's heart, we have the right side in blue and the left side in red. And we're going to have to deal with this being a multi-scale problem, a multi-modality problem, as well as a multi-physics problem. So when we think about the way this works, and particularly when we're talking to drug companies or people developing therapies, we get a lot of information in the clinic, which is often describing the whole heart, the heart at the organ scale, or integrated measurements that might be pictures of the heart from MRI or echo or CT or ECG measurements or circulating troponins or BMP, and they provide very much an integrated understanding of how the whole heart is working. Yet when we talk to people who are developing therapies or trying to understand disease mechanisms, often they're working at kind of the cellular scale. They might be looking at the myocyte. And so how do you start to link these cellular scale pathways and function that you have at this much smaller scale through to this whole heart function? We also have an awful lot of data about our patients, and this is kind of classic data from CT or MRI or pressure measurements and genetic tests, histology, demographic data, but increasingly we have kind of wearable sensors or implanted sensors that provide us continuous information on tracking patients. I don't know if I'm supposed to be reading messages or not, but I will assume that I'm not. If people want to jump in, feel free to speak up. And we have a lot of data, but obviously we're all in different hospitals and everyone's experience will be different. But in the hospitals I've worked in, we have kind of this vision as to what the data management system should look like. And that we should have this beautiful representation. It should be like Minority Report. It should be fantastic. And yet, when we try and find data within our hospitals, often it looks a little bit more like this. And I say this from working in cath labs, where if you ask for a particular patient's data, it isn't stored on a PACS server as you might with an image, but it's going to be stored on a DVD stored somewhere at the back of the catheter lab in a non-chronological order. And so managing how we integrate all this data is one of the other challenges within the hospitals. And they also have multi-physics systems. And so when we're thinking about the heart, we're trying to combine information about the chemical side of the heart, the metabolism, about the electrics, about the mechanical elements, and also about the fluids. And how do we represent all these different systems and their feedbacks between each other within one system? And so we'd like to think about then how we can make these mathematical models of patients. We want to think about how we can use them to guide therapies as well as predict outcomes. And when we're thinking of predicting outcomes, one of the examples that most of us have interacted with will be that of the weather. And so this is a physics-based system, a digital twin, if you will, of the globe, and it's trying to make forecasts of what's going to be happening in the weather going forward. It's going to integrate data from multiple different scales all together and then make these predictions for us. And so sometimes it's worthwhile contemplating what it means for making a digital twin of a person versus these kind of similar digital twins or mathematical models of the weather system in the globe. And so this is as a parent, one of the molecules that I'm very, very familiar with, and this is our good friend, the caffeine molecule. This is about 500 picometers across. And so when we're thinking about the degree of multi-scale problem, we're thinking about drugs that might be interacting with the heart. What are the different length scales that we're going to need to capture in our mathematical models? And so you start by taking your molecule in, someone who's about 2 meters high, going across the size of the heart at 10 centimeters, across the myocyte at 100 microns, and transitioning across the cell membrane at about 5 nanometers. It's binding to molecules, which then change the calcium release inside the cell structures, binding to the sarcomere at 2 microns, changing the movement of blood cells, causing a change in heart function, and then altering the overall cardiovascular function. And so this means that when we're looking at trying to simulate something from a molecule like a drug to its effect on the cardiovascular system or the whole body, we're spanning about 1 to 4 billion length scales. So we're trying to say how something that is 4 billion times smaller than the body is affecting it. And what does that mean analogously for the problem that we're dealing with when we think of the size of the globe, if we put that in the context of weather? So as my accent would suggest, this is how I view the world. We're about 13 million meters across. And so what will be 4 billion times smaller? And if we zoom in on New Zealand, that's about 1.5 million times smaller. And if we zoom in further, and if you spend enough time on Google Earth, you will invariably find a picture of yourself. And so I'm here at about 1.8 meters high with boots on at about 650,000 times smaller. And the glasses I'm wearing in the picture are similar to the ones I have on today. And they are about 3 millimeters across at the lens. And that is what is 4 billion times smaller than the globe. So when we're thinking about making these digital twins and mathematical models of patients' hearts and cardiovascular system, we need to be thinking about how do we make a mathematical framework which can link something which is which would be analogous to linking something that's 3 millimeters across to the global weather patterns. So how do we do this? And so we break this problem down. And so we start by simulating the electrical activation pattern. So we can simulate the electrical activation propagating over the right atria before activating the left and activating the left and the right ventricles. That electrical signal, which has been carried at the whole heart scale, is then represented at the cell scale by ion channels or proteins, which span the cell membrane, modeling the movement of ions in and out of the cells. This links to a chemical signal in the form of calcium regulation in the cytoplasmic reticulum inside the cells, which gives a calcium transient, which then binds to the thin filaments, allowing the thick filaments to bind within the sarcomere to generate tension. This is all encoded within a tissue matrix where we're able to represent the preferred orientation of myocytes and collagen structures to represent preferred stiffness, conductivity, and active contraction. We can then combine that tissue model with a representation of the boundary conditions on the heart, capturing the preload and the afterload from the cardiovascular system, as well as the effects of the pericardium, allowing us to simulate mechanics. Then we can link that wall motion from our mechanics model to the Navier-Stokes equations, simulating fluid dynamics, allowing us to model the blood flow in and out of the heart. So this provides us with an end-to-end simulation platform, which starts at one side with our representation of the electrophysiology, which we can measure, and goes to the other with the fluids and motion that we can measure as well from imaging technologies, and allows us to understand how the cellular structures in the middle interact between these two systems. Now, this would be a representation of a model of a single heart, and this was certainly what we spent an awful amount of time trying to produce. But we'd also like to make a model of an individual's heart. So how do we go from the abstract representation of a heart to modeling a particular patient's heart? Now, the challenge we have here is that patients don't come with plans. And so unlike other engineering applications where we might be able to download the original plans that we use to build the bridge or the aeroplane, we have to make our plans for ourselves on each patient. Similarly, in healthcare, unlike in other engineering applications where I can go and look up to find out how strong steel is or how tough aluminium is, the patients we have don't have material properties that we know, and many of them are not new builds. And so they don't have shiny new properties, and I will very much include myself in the kind of renovation body. So we have to work out what the material properties are for that patient, and by the time they come to see us, they tend to be unwell, and so there's lots of heterogeneity in those material properties. So how do we do this? We start with we're building our mathematical model of our patient on a computer, our reference model, and then within this we encode known physics, so conservation of energy, conservation of mass, conservation of charge, and we can add in biological elements as well within this. We can then take biological measurements. These might be measurements that have been made on ion channels or on cross bridges, tissue structure, and we can use machine learning to embed that information within our mathematical models. We can then take clinical measurements. This might be from MRIs, wearables, or echo, and we can then again use machine learning and AI techniques to update the model to personalize it to the individual. And once we have this personalized model, there are two things that this allows us to do. One is prediction and the other is inference. So for prediction, what that means is that I can have someone come and say, I have a new drug. I want to simulate what effect does lidocaine have on this particular ion channel, and what will that look like as far as its effect on the ECG. So I can take our mathematical model, I can alter the effects or the effect of a drug on an ion channel and predict what effect that will have on whole heart function or on the ECG of a patient. The other thing I can do is inference. So I can have a whole load of clinical measurements from a patient, MRIs, pressure measurements, and I can then infer or estimate what the material properties are. So in heart failure, that might be saying how hard is the heart contracting, how stiff is the myocardium, what is the aortic resistance to blood flow, and that provides new endophenotypes that we can use to characterize a patient's disease, how that disease progresses, and if a patient is responding to therapy. And in this way, we can start to use the models to inform patient care. So when we're building a mathematical model of our patients, at the moment, as I said, we're very much in the research phase. And so we collect an awful lot of measurements. We get CT data, MRI data, we'll get electroanatomical mapping data, ECG data, as well as pressure measurements in all four chambers of the heart. And we can then also collect these measurements under different pacing conditions and at different pacing rates to provide a very rich data set to constrain our mathematical models. We can then segment out the heart, and this is where we start to kind of use AI methods which are automatically trained to label different regions of the heart. We can then automatically mesh that heart to create a mathematical description of the patient's anatomy, not just a label from an image. And then we can introduce representations of the myocyte orientation in the form of fibers in the left and right ventricle, as well as the atria, derived from either cadaveric or DT-MRI datasets. So we end up with a computational framework. This will have a mathematical model which represents about 81 different proteins spread between the atria and the ventricles, representing the simulation of the transmembrane potential, the calcium transient, and tension generation in the myocardial tissue. We end up with about 15 different properties that characterize the tissue. So that's the conductivity, the stiffness, the pericardial boundary conditions, and about 21 different properties describing the circulatory system. So overall, we end up with about 120 properties, which we can then use to describe a patient's heart, and these provide the kind of endophenotypes that we can then look to infer. And the result is a heart model which can simulate all of the measurements that we're kind of making clinically. So this includes the pressure measurements, the volume measurements, as well as the 3D activation pattern measurements that we can infer with either ECGI or electroanatomical mapping data sets. Sorry. And so we then end up with taking this piece of information. We can then look at what parameters we can constrain. So here we've taken a patient who's had the pressure measurements, the ECG, and the CT measurements. And we can look at constraining about 25 of those 120 parameters. So we don't have measurements which can describe all of them. So we can only characterize a subset. But here we have parameters which are kind of referring to aortic resistance, pericardial stiffness, sarcomere tension. So a number of measurements which are going to relate down to the molecular structures that we might be interested in characterizing a patient's disease or be using to how we can make a prediction on the response to a therapy. So this is how we can take these large data sets, these mathematical models, and tune them to represent individual patients. So one of the applications for this type of approach has been the patients with desynchronous heart failure. So patients who have a healthy activation pattern, their atria rapidly activate, then the left and the right ventricles activate before, and in patients with synchronous heart failure, you'll often see a left bundle branch block where the left bundle of the Purkinje network fails. There's a slow activation from the right ventricle to the left ventricle, and that will lead to a desynchronous, inefficient, and ineffective contraction. You can then look at placing leads into the left and the right ventricle, and this will augment that activation pattern, returning the heart to a synchronous contraction or activation pattern. And so one of the areas that we were interested in was our clinical colleague came to us and said they were going to use a new system, and this is a leadless pacemaking system. So they have one device which is pacing the right ventricle. They have a pacing electrode, it's about 9.1 millimeters long and 2.6 millimeters across, which is going to be implanted on the left ventricle endocardium, the inside of the heart. And they have a battery which is placed outside the rib cage and an ultrasound transducer which is placed between the ribs. This ultrasound transducer is then going to provide ultrasound energy to the leadless pacemaker, which converts that ultrasound energy into an electrical stimulus, activating the heart. And the problem they had was that they wanted to implant everything all at once, and that meant that you needed to ensure that the ultrasound transducer was able to deliver energy to where you were going to plant the electrode, but the way they were doing it was implanting the transducer and then working out where they could put the electrode afterwards. And this led to some mismatches between energy delivery and energy requirements, which was causing problems for the patients. So what they wanted was a pre-planning system where they could put all of this information together for a patient and plan where they were going to put everything before they did the original procedure. So for this, we could look at using machine learning methods to segment and then track the CT motion, allowing us to work out where late contracting regions were. We could do perfusion imaging with CT to work out regions which weren't receiving a lot of blood, potentially infarcted. We could look at the electrode, and it has a 3.6 millimeter long barb, which is going to be plunged into the wall of the heart. And so we wanted to make sure that there was going to be plunged into a region which was not too thin. So we wanted to make sure it was at least 5 millimeters or thicker. And so we could make a wall thickness map. And lastly, we were able to make a mathematical model of the patient's heart representing the electrophysiology, tuning that model to the individual patient, and then simulating what effect pacing from each different location would have on that patient's activation, try and work out the best location to pace their heart from. We'd then present all of this information to our clinical colleagues before a procedure and make a ranked list of the regions of the heart where we might want to put the electrode. And so the way this worked was we would have patients would come in and have a CT scan. We would make our model and work out where to pace the heart from, or predict where to pace the heart from. We would then register that model with the fluoroscopy that was being recorded during the procedure. And this was a very important step, so that instead of making a digital twin or a model in a computing lab and doing this in isolation, we worked with Siemens to look at how we could put this information in front of the cardiologists, and so that it could be provided to them in a way that was going to be that they could use in the procedure. And then we would have these regions of the heart, the AHA segments, which were the target locations, and they would then be able to guide the catheter to that location to deliver the leadless endocardial pacemaker, the target location that we were aiming for. And so this was a way that we could have patients come through, make mathematical models, work out where we should be delivering that therapy, and then put that in front of the clinicians to evaluate how effective we were at identifying the optimal place to pace their hearts. And so we went from this idea of having patients come in with a conventional workflow where they would have a measurement made and a treatment decided, so that they would instead have measurements made and a model made, and then we'll be able to make a fancy model that would then be able to kind of be used to inform their treatment. But what happens is, in realizing that we could do this faster, instead of making one model, you can start to make lots of models. And so you can end up generating virtual patient cohorts. And once you have a virtual patient cohort, you don't need to necessarily simulate just the device that that patient was going to receive or did receive. You can simulate any device on them. And this then allows us to start to do in silico trials where we compare the therapeutic outcome or predict what the outcome will be for multiple different devices to work out which ones will be best in which different patient groups. And so with cardiac synchronization therapy, we started off just with biventricular epicardial pacing. Then we ended up with biventricular endocardial pacing. More recently, we've had the HIS endocardial conduction system pacing, left bundle branch conduction system pacing, HIS bundle branch with an additional left ventricle lead, and left bundle branch with an additional left ventricle lead, and the list goes on. So as we end up with a family of different systems for delivering cardiac synchronization therapy, it becomes harder or becomes more important to choose the right system for each patient. But it makes the trials and designing these studies more complicated because there are so many different options you could have in each patient. Do you need to recruit more patients? Do you need to spend more time delivering different therapies in individual patients? What does this look like? So what we've been able to do is to take our virtual patient cohorts and then look at simulating all of these different therapies in a standard cohort. And so we can simulate left bundle branch block, epicardial pacing on the lateral wall, endocardial pacing on the lateral wall, septal pacing, HIS bundle, left bundle pacing, all these different permutations. And the doctors keep coming along, as well as device companies, asking us to simulate more and more different combinations. And what this allows us to do is to make predictions before the clinical trial. It allows us to better understand what type of patients may be more or less likely to respond to therapies. It also allows us to understand how device settings may lead to unexpected results that wouldn't have been known until the trial was started and the protocol was designed. And the other thing we've been doing for our clinical fellows is the clinical fellows, and particularly in cardiology, will often need to apply for funding to do a PhD. And so we will provide pilot data for their fellowship, which then can use to justify doing the prospective clinical studies. And so we were then, having made some of these model cohorts available, we were contacted by the FDA and they asked us to create some additional cohorts. And so we're now looking at publishing models which are both male and female. So we have sex balance between our models, represent different severities of heart failure, and also represent control cases so that we can make these as kind of a digital asset for device companies or people wanting to do clinical studies in virtual cohorts. And so having looked at these kind of simulations, we then started saying, what are some of the questions we can ask and how can we scale this up? And so one of the questions with CRT was, or cardiac synchronization therapy, was deciding who should get a pacemaker. And when we look at the guidelines, we can see that there's multiple numbers with different levels of support based on if you have a left bundle branch morphology or not. But the one thing that is clear here is there is one number for everyone. It doesn't matter what your ethnicity is. It doesn't matter what your gender is. And yet we know that women are more likely to respond to CRT than men. And one of the reasons that it's possible that this may be, it's a hypothesis, is that women in general have shorter cure durations than men, potentially because they have smaller hearts. And so one of the questions that we had was, what is causing these 6 differences in this QRS activation duration? And is it due to a difference in heart size or a difference in material properties? And so can we look into that? And should we be considering this when deciding who gets this therapy? So for the QRS duration, we can measure the ECG. The QRS duration is this big R wave here. It represents about the activation time for the ventricles. And it is likely due to properties of the sodium SCN5A, or the sodium channel, as well as the connectivity between the cells and the fiber burden, but also due to the size of the heart. So a bigger heart, if everything else is equal and the activation wave has the same speed, will take longer to activate than a smaller heart. And so which of these factors are important? So for this, we could look at a UK Biobank. And so here we'd started to get some confidence because we were making models of tens to hundreds of patients. UK Biobank provides us with a population level data set, which allows us to make broader statements. But when we started this project, they had about 56,000 MRIs and similar number of ECGs. And they've now moved that up to I think they're getting very close to their 100,000 MRI target. Now, for the in silico trial work, all of this has been done on up to 100 cases so far. And so we had to really think about how do we apply the workflows we've made for doing relatively modest clinical trials, how do we scale that up to these kind of large population cohorts? So here we could automate the segmentation process. So we could automatically label different regions of the heart, of the images corresponding to different regions of the heart. We could automate the process of extracting out a 3D surface mesh, making a mathematical description of that patient's heart, adding in some heart-specific coordinates so we can define stimulation or activation patterns in all cases, and defining material fiber coordinates. So this meant it took us about 5 minutes per case on a standard desktop, and we do 500 cases per day. It's worth noting that at 500 cases per day on a reasonable desktop, that's still going to take 100 days or 3 months to do 50,000 cases. So working at the scale of populations for these approaches has some significant computational costs. What we wanted to do here was, in part, was to characterize what is a representative heart for the different types of people in UK Biobank. So having made 55,000 models, we spoke to UK Biobank and asked them if we could publish them. And they said yes, but only within the UK Biobank space. And we said, well, can we publish average hearts? And they said yes. And so we agreed that 3 or more hearts averaged together would be an average heart. And so we could look at the distribution of male versus female, at different ages, at different BMIs, and look where we had different permutations where we had at least 3 hearts or 3 individuals in each of those bins. And then we were able to make 1,400 different cardiac atlases, which we then provide as a data asset, which is a representation of all these different age, sex, and BMI hearts that people can then use for their own simulation work for balanced sex and age distributions. We then want to say, can we then use these models to start looking at, can we infer material properties, or can we kind of derive something more about these hearts than just the information that's in the images? So we could take the MRIs and the ECGs. We could have our mesh, which gives us a characterization of heart size. We could then run a simulation on each patient's heart to estimate what the activation pattern will be like, to simulate the 12-lead ECG. We could then compare the simulated 12-lead ECG with the measured QRS duration and use machine learning to update what those material properties are in that patient's heart to get an estimation of what the conductivity in that patient's ventricle will be. And again, this is not a small task. It takes about 100 cases per day. So in this case, we were only able to work with about 3,000 cases or 1 month of compute. But what we end up with are these studies where we can start to make mathematical models of about 3,500 people. We have balances between male and female hearts, and we can look at the QRS duration, which is in red, which is the full distribution. So we have a wide distribution for these different groups. And we see a significant difference between the male and female hearts, with the female hearts having a significantly shorter activation time. However, and I was surprised by this, was that the conductivity that we saw was not different between male and female. And when you end up with 1,700 to 2,000 cases, you can have very, very small differences in the mean and still get a significant or statistically significant difference. But we saw no difference. And we were able to explain the entire difference in the QRS duration activation time by the myocardial volume as opposed to the conductivity, where we saw this significant difference in myocardial volume between the male and the female hearts. So in this sense, if we were thinking about indexing the ECG, or the QRS duration for deciding who should get a therapy, we would want to be thinking about how we can index that ECG to the size of the patient's heart. And we possibly don't need to consider their conduction properties. Now, the one thing we have learned, and this won't be a surprise to anyone, is that patients don't live in the hospital. And so this is really what's been driving us to think about making digital twins. So all the models I've shown you so far could be described as patient-specific models. And we are trying to look at how we can then, and these models represent often a patient having a procedure or lying in an MRI scanner. But the patients we have are going to be out in the community living their lives. And so how do we track those patients as they're out in the community from sensor images? So with digital twins, we can look, the particular group we're looking at are pulmonary arterial hypertension patients. And working with people up in, or Alex Rothman up in Sheffield, he is implanting CardioMEMS devices in the pulmonary artery. And so this provides us with nightly updates of the pressure in the pulmonary artery, as well as having annual MRIs for these patients. What this allows us to do is to make again, using machine learning to update a mechanistic model representing the cardiovascular system for these patients. So this allows us to estimate what the parameters are through time for these patients. And what we end up with are these plots. And so I need to really update this figure, but this is 2 years of data. So this is a patient who's had a CardioMEMS device and has been updating their pressure transients every night for 2 years. And the red line was when they started a new medication. And we can have all of these endophenotypes, which are inferred from their pressure transients and their volume measurements, and look at how they track through time. So we can make estimates of what the blood volume was, what's the systemic arterial resistance, which we see no difference in the pulmonary arterial hypertension patients. We see a very rapid decrease in this patient from their pulmonary arterial tree resistance, and a much slower, possibly remodeling change in their arterial tree compliance, along with significant reduction in the right ventricular active contraction. And what's interesting here for us was not necessarily was about the ability to monitor patients to better understand how drugs might be mechanistically affecting patient physiology, but also the ability that if we can repeat this on lots of patients and have a better understanding how drugs are affecting patient physiology, of having earlier markers of when a drug is working or isn't working. So for some of these patients, they might take a drug and it might not be working on the particular mechanism which is driving their pathology, and so they won't see any improvement. In that case, we should be looking for a different therapy. Conversely, there will be other patients and potentially that drug won't be being metabolized correctly, and so they won't actually be seeing the active ingredient, or that drug might be metabolizing too quickly. So we have the correct mechanism, but the drug isn't working for other reasons. And so understanding if the drug is having its correct mechanism of action is an interesting way to think about how we can better identify if a drug is working or if we've identified the correct mechanism in our patients. So this was the work that we've been doing, kind of spans a number of years, and looking at how we can make mathematical models of patients' physiology, looking at how we can capture the shape of their hearts, the material properties of their heart, simulate their therapies, simulate them through time. And our goal is to really provide effectively a computational model both to patients as well as to doctors, and they can play a computer game of trying to cure their heart or improve their heart, where they can use as many lives as they want, learn as much as they want about their heart, and hopefully mean that when they deliver therapy, it's the right therapy at the right time for the right patient. And this was just to acknowledge that to do this, we have quite a large group and a very large kind of collection of international collaborators, and particularly supported by people at Turing and Imperial to deliver this. And so for that, I can hopefully someone's been reading the chat, because I have not been able to follow chat and speak at the same time. So I apologize for that.
Huge round of applause. Obviously everyone's at home, but you can see, I mean, look at that. Yeah, it's quite effective how Zoom does that, isn't it? Lots of appreciation for your talk, Steve. Thank you so much. I mean, that's really, really fascinating. One question that did come up in the chat and you know, came to me as well, which is, how far away do you think we are from being able to use this kind of digital twin technology in routine clinical practice?
I mean, I think there's an interesting question about what is routine clinical practice. So I think one of the reasons we started looking at the pulmonary arterial hypertension as a patient group was they're served by, I think it's 12 centers in the UK. And so you can have a national impact with a much more finite number of kind of participating centers. They're also a patient group who have very expensive medication and have a kind of, at the moment, they're having kind of devices and sensors implanted in them to help them better manage their care. So I think where you have patient groups who have sensors, who have implanted, who have expensive medication, are treated at very specialist centers, then we're actually quite close to looking at how we can translate this work into clinical practice. And certainly the program grant that I wrote and the promises that I made in that program grant would suggest that in 4 years we will be delivering this as an NHS care pathway. We will see how much progress we make in that ambitious goal, but that's the kind of timeline that we're looking at. And so we will be looking at, in the next 6 to 12 months, starting to run kind of observational studies, looking at doing the feasibility of how we start to move the data from patients into a trusted research environment, how we start to make forecasts for those patients, and then look to see how they match with the patient outcome. And then, while also at the same time interacting with clinicians to understand how they want that information presented, talking to patient groups about what is acceptable to give to patients, and that we don't want to be making very speculative forecasts, which may be inaccurate or wildly inaccurate, not insufficiently accurate for patients to make decisions, and are just more concerning. So there's a lot of ethical and operational questions that also go around the technical and bringing this into clinical practice.
Yeah, that's interesting. That particularly, it's particularly interesting to think about what's most useful as the output from these models.
Yeah.
And you showed those amazing, videos showing the effect of the different types of electrical stimulation on the heart. Do the clinicians that you've worked with so far like to be able to see those video visualizations of the different ways the heart moves, or do they prefer some kind of output? The model kind of saying, just use this particular method.
So with the clinicians, what we've often been doing is evaluating the clinical trial designs that they're working on. So they will come in and say, we have a new device. We're going to see if it's any good. We could tune the delay between the atrial lead and the ventricle lead, or between the two ventricle leads. Do we need to do this in our protocol? Is that likely to have a big impact?
Yeah.
So we will then run a series of simulations, show them the videos, and that's, I guess, largely it gives them confidence that it's where the numbers come from.
Yeah.
And I think it speaks to that kind of explainability question that you often have with AI, where if I can show you a model and it's activating and it looks like a patient, and it kind of, you kick the tires and it behaves the way you expect, people are a lot more confident in that than they are in my random forest model says that this patient needs this result.
Yeah.
And I appreciate that there's ways around that, but that's kind of where we've got to. So a lot of our work is using AI and machine learning to take data and put it into our models, and then taking results from those models and presenting them to clinics, but it's all centered around this very much physics and physiology informed simulation.
Yeah. And you've got, incredibly rich data sets that have gone into creating these models. Emma McMillan, are you there? Because you've asked some brilliant questions in the chat, and I just wondered whether you wanted to unmute and ask them in person.
Yes, I am here.
Hello, Emma. Welcome.
Thank you. Sorry, let me leave the library.
Oh, right, okay. No problem.
Yes, hi. So thank you for the great talk. It was really interesting. And I saw that you also did your DPhil at Oxford, which I'm doing right now, my DPhil at Oxford. So yeah, I work in the computational cardiovascular science group. So I work on basically 3D mesh generation from MRI. So I had a lot of questions sort of about your integration of the sex differences into your models. And from the UK Biobank, I don't know exactly the percentage between like men and women in the data set. But yeah, I was wondering how you sort of propagate the error between the multiple steps in your pipeline, because you're going from like segmentation to 3D mesh generation, then to this ECG prediction. So yeah, how do you make sure that your error is sort of staying within a reasonable bounds between each of these steps?
Yeah, I mean, there's two parts, I guess, to that question, or there three. So the first one was, I think it's pretty 50-50 for UK Biobank. I think there's slightly more women, but we're talking very, very minor amounts. What we've done for the kind of error measurements is we've done the standard kind of methods of train, test, and validate with a split of different groups, and then cross-fold validation to kind of evaluate with different measures of Honsdorf distances and dice scores and what have you for evaluating how well the segmentation is working. So we can have some measure of, is the segmentation working? We've followed kind of relatively standard practice for that. The meshing, what we can do is we can make the meshes and then we can reconvert those meshes back to an image and then look to see if the difference in the image generated from the mesh, how similar is that to the image that generated the mesh in the first place. So that also can give us a measure of any error introduced there, and also doing projection of the mesh back onto the labels and making sure those distances are relatively small. So those are the kind of segmentation, meshing. The calibration method, what we can do there is with in silico verification approaches. And so what we can take is simulating a result where we know what the ground truth is, and then we can say we're going to throw away the parameters we use to generate that ground truth, and then try and recover the parameters using the simulated result that we have. So that means that we can kind of give some estimate in an ideal situation with no noise, with no model discrepancy, how well is our fitting method able to recover the conductivity if everything is correct. So in no way have we covered everything, and in no way can we say with certainty that the conductivity we have is going to be precise for each patient. The other thing we've done to kind of reduce some of that is, if there is uncertainty, and so long as that uncertainty is in either direction, we've done this to a large number of cases. And so that at least reduces some of the kind of random distributions from uncertainty, as long as we're not biased, that allows us to cover some of that uncertainty due to just random noise in the processing. So those are the multitude of ways we've done, but there's invariably ways that you could do that better, and we could also look at how we could encode uncertainty and propagate that uncertainty forward in each of our predictions so that we could then represent that uncertainty more explicitly, but we haven't done that yet in this case.
Thank you. Yeah.
Brilliant. Thank you so much, Emma, for the question, and Steve for the answer. Venet, and also Catherine Upex, if you were able to ask your question in person, it'd be great after Venet. Venet, far away.
Yeah, thanks for the great talk. I mean, I was really enthused by the fact that, you're using this kind of, not only multimodal, but multi-scale data. And I think, this is something that definitely we need very much, not only for digital twins, but I think in modeling in general, because otherwise we are just not capturing the patient's state. But of course, one of the issues with kind of, multi-scale and multimodal is the sparsity of data, because you very rarely get, the across scales and across modalities for a single patient, let alone when you go to kind of, population level. So I'm just wondering how you dealt with that issue in your modeling, and perhaps if you're considering anything around synthetic data as well.
Yeah, I mean, it's a great question. I think at the multi-scale, it's challenging, particularly in the cardiac space, because most patients are not getting biopsies. Even when you get a biopsy, they are never as big as you would want. And they're never you never necessarily know exactly where they're taken from. And so there's lots of issues there. What we are doing is we're working with people who have, I guess, rich data from explanted hearts. So these are people who are doing spatial transcriptomics from tissue across the heart using explanted hearts. They have a lot of tissue, and that's providing us with distributions of mRNA across the heart. And so while not patient specific, it's providing us with priors or kind of rough estimates of what these values would be. And we can use that as an initialization or as default parameters if we don't have enough data to try and tune the models to individual patients. So that's how we're starting. It's, again, I'm very happy to acknowledge that is a imperfect approach, But I look at the cancer people with all of the tissue that they seem to have and all the spatial transcriptomics they get with a great deal of envy from a science perspective. So in cardiac, we have to work with what we can. I would also say that the imaging data, though, is improving. So photon counting CT gives us a step change in resolution. I think that's a really exciting and interesting area to look at. And our MRI colleagues are continuously coming up with improvements in things like diffusion tensor MRI resolution and different measurements, T1, T2, what have you, which we can then add in another layer of tissue characterization into the models. For synthetic data, we we have generated synthetic data from the simulation side, which people do use, but that we don't tend to use. We're not overly reliant on synthetic data at this stage in our model creation, but it is something certainly we should be considering.
Thank you.
Brilliant. Actually, just one quick question, and this can be as short as you like, Steve, but are there any other organs that you look at and think, gosh, I wish I was digital twinning those instead?
I mean, from a funding perspective, no, because it's not a terrible space. But I think there are certainly options or opportunities in the respiratory side, and that's something we'll be looking into. I think that kind of kidneys, livers, there's a lot of work done on musculoskeletal. So I think that there's a lot of areas where there is added value. But often it's just the infrastructure and the data needed to bring this together in a sustained way is quite challenging to maintain without kind of a very reliable funding stream, which we've been very grateful to the British Heart Foundation for supporting this.
Yeah, well, I'm very deserved as well. Catherine, thank you so much for joining us.
Hi, yeah, thank you for a really interesting talk. I was just wondering, I'm a biologist, biomedical, I come from that kind of background, and I was just wondering how much these models can be informed by more of the discovery science side. I've got a lot of colleagues that say like work on the hERG channel and blockers and discovering new blockers for the hERG channel and kind of how it affects the trafficking of these ion channels and things. And obviously that's like on a very small scale. But I was wondering, do the models replicate kind of the in vitro, in vivo findings? And are they flexible to be updated based on that? Or is it not really at that? I guess a lot of these discovery science things are at a very like primary kind of stage, but are they flexible to be updated as the science progresses on that side?
I mean, absolutely. So these models started. We can have a debate about this, but starting something is always hard. But about in the 1960s with Dennis Noble at Oxford, and he started with modeling the hERG channel and looking at modeling the Purkinje myocyte. And so those same cell models, which are being used by the FDA through the CEPA initiative, which is about kind of looking at thorough QT studies and replacing them or augmenting them with stem cell work as well as single cell simulation work, we use exactly the same cell models and we put them into our whole heart model. So if you came to me and said, I have a new, I've done some expression of an Xenopus oocyte, I put a bunch of drugs on, I have got a genetic mutation or what have you, I can tell you what the kinetics of the hERG channel are, and we can make a mathematical model to describe the genetic, the drug, the whatever changes in the hERG channel kinetics. I can immediately take that and put that straight into my whole heart model, and we can look at what effect that has both on the ECG, but also what effect it has on the calcium dynamics and what effect that has on the mechanics of the heart as well. So you can kind of link all of that together. And then more recently, we're looking at how you can simulate sex hormones or sympathetic nervous activation, and how does all of that feed in, particularly when you're thinking about hERG channels, to the wider kind of cardiac function. So in short, yes, and the cell models are very cheap to solve, so it's very easy to expand them out to make them more complicated.
Great, thank you.
Brilliant. And so I guess Catherine's question is how, looking at how you take developments in knowledge and put them into your model. Lewis asks an interesting question, which is, have you been looking at the model and then seen the outputs from it and noticed any discrepancies or unexplained differences that point to new discoveries so that are achieved through taking this approach?
I mean, there are.
You may not want to share them.
No. That's fine. I mean, a lot of it is about, so often a good modeling question is one where there are three hypotheses from three different labs, all publishing lots of evidence entirely independently and not talking to each other. And then we can take all those three hypotheses and put them into one common system and say which of these are plausible or not. And so that is a way in which you can kind of start to differentiate biological elements. Other times we will put things in, and so the CRT would be another example. We made our patient-specific heart, and we thought, what determines who responds to cardiac synchronization therapy? And one of the things we predicted, and we're still looking at, was that the Frank-Starling mechanism, or the length dependence of tension. So as you pull muscle, it generates more force. And so in CRT patients, it looked like patients who had a functioning Frank-Starling mechanism were less likely to respond to therapy than those who do. And so that was a prediction we made from the model, we published it, and now we've been looking at how we can then kind of clinically measure the Frank-Starling mechanism effectively in patients' hearts, which is not as easy as you might expect.
Brilliant. Steve, thank you again so much for joining us today. Thank you for your brilliant presentation. Thank you for your amazing answers to some really quite diverse questions. And it's been an absolute privilege to have you join us today. Thank you. We're coming into land, so thank you everyone for joining. And anyone who's able to, please do come along and join us on the 11th of November for our next monthly meeting. And that's with Jeff Hogg from the University of Birmingham, looking at the AI readiness checklist. Until then, all the best. Thank you so much.